So it's very nice to be here. And if you're curious, you should take a look at the March issue of the Nature Methods, where there is not only the paper which you're going to show, but also the papers talking about this technology, so the DNA sequencing. But the problem that we're going to address today is transcription factor binding. And the typical approach to do this is, of course, to have sequence-based methods. For example, that you have logo representations of how one particular protein is binding to DNA, and then you scan genome-wide, usually for some reason upstream of the gene, to trying to find these transcription factor binding sites. But the problem is that these motifs are small, the regions you're looking for are very large, so in the end you end up with many false positive predictions. And at least in higher eukaryotes, the issue is that there is no such a thing as a naked DNA. Actually, DNA is wrapped around the chromatin structure, and the chromatin structure has two main structures. So this condensed chromatin, where actually it's wrapped around these proteins called nucleosomes, and actually no binding event can happen. And even in the case where you have the open chromatin, and this is when actually binding is possible to happen, there's still some histones around. So actually, only particular regions of your DNA, the transcription factor binding will happen. And so actually, luckily we have nowadays methods to measure these things. And I'm going to talk about one of them, which is DNase-seq. So the idea with the DNase-seq experiments is that you first freeze the interactions of your DNA and your chromatin. Here you have a particular some histones and some transcription factors. And then you put DNase, and DNase is an enzyme that will cleave DNA wherever is unprotected, so where there is no binding events. And this is usually happening between transcription factor binding sites. So out of this, we're going to have now these fragments of DNase and your proteins. You isolate your RNA. Depending on the protocol, we either get only small fragments or only the beginning of these DNA fragments. You can sequence, map back to the genome, and then you should have a signal like this, where you have your reads should be aligned to the region where, let's say, you have active binding sites or open chromatin. But actually, if you do the right signal processing, which is because the cleavage event here happens exactly in the start of your fragment. So if you only count the start of your reads, you should see also this kind of signals here, which we call footprints. And the footprints are reflecting the fact that if you have a transcription factor sitting here, it will protect the DNA, and it should have this kind of values between high levels of DNase. So one of the tasks that you want to do is, in particular, is to detect these transcription factor footprints. And not surprisingly, this kind of methodology was explored in papers like one of the main papers in ENCODE, where they say that, for example, now they could find the whole regulatory lexicon in mammalians. But in the last years, there have been a few words discussion, actually, if some of the results on the DNase. And one of them, here at all, they showed that actually there was an issue. So if they did an experiment where they have unprotected DNA, they put the DNase, and they saw that there was a bias in the cleavage event. So that particular sequences have more cleavage cuts than other sequences. For example, in this case, here we're more likely, 100 more times likely to see these sequences than these ones. So, and if you're actually trying to find, you could try to estimate bias estimates, which is kind of how many reads you see in your experiment versus how many you expect from your genome. You could try to get profiles like this. So here is an average profile of the DNase around p53 chip-seq peaks, which also have a motif, which is associated to p53. And what they show is that the bias estimates were actually also having a footprint. So this was just an artifact of the fact that you have a sequence conservation around your transcription factor binding sites. Another issue that was also shown by Sung and colleagues is that transcription factors, they have different binding time. So CTCF, for example, is a stable protein binding. And actually, when you look at the average footprint profile around CTCF, you see what they call the deep protection. So the difference between the footprint signal and the flanking regions is high. And this is different from things like nuclear receptors, like as the GR, which has a very short binding time, meaning that in the end, in the populations of cells that you're measuring, which are usually millions, only a few of them are likely to have the event. And as a as a result, then the average footprint profiles are very slight, so they will have very little protection. So actually they claim that it's very hard, or it's possibly likely that you cannot really detect this kind of factor events. But so what we did was we tried to revisit the problem, and the first thing we did was we looked at the literature, all methods which are out there, to try to find these footprints computationally. And there are two main classes of methods. One is segmentation-based, so the idea is simply that they have your DNase-seq signal, and then you try to find for these footprints, so these values around these high peaks. And there is another class of methods which works a little bit different. So these ones, they usually start with a motif. You look in a genome for this binding sites with these motifs, and then you try to classify all your hits that you have of that particular motif by the footprint shape around it. So for example, if you have a valley, then this should be a bound. If you don't have a valley-like, then you should be unbound. And yeah, so but the other question is then how we evaluate this kind of data. And this is actually how the footprint predictions for segmentation-based method looks like. So what we have in the end is simply these blue regions here, which are somehow supported by this DNase value. So everything what we do is we filter the genome and say, okay, binding sites should be only happening in these blue regions. But to really capture back what is the binding sites behind them, then we need to look back in the sequence. For example, we can do motif match here for a few selected factors, and then we can ask things like this. So if you have a good agreement with the footprint and your motif, then this is supposedly a footprint, a motif that's supported by this footprint, so it should be active in these cells. You can also have, for example, for the same factor, for CTCF, motifs which are not supported by footprints, so these are the non-active binding sites. And one way you can evaluate this is you can use now ChIP-seq of the transcription factors, and now whenever you have, let's say, your active predictions which also supported, this will be your true positives. On the other hand, your negatives, which are also not supported by ChIP-seq, they will be your true negatives. And out of this, of course, you can measure sensitivity, specificity, and error under the curve. Okay, but before I show the results, I have to talk a little bit on how we can try to deal with the bias estimate. And the way that actually we and most of the people do now is you use a k-mer, and this usually 6 mers, and then you kind of measure how many reads line to the middle of the 6 mers versus how many 6 mers we have for that particular 6 mers in the genome. But there are actually two strategies how you can measure this. One is by using naked DNA experiments. So these are DNase experiments where there was no chromatin around. But the other approach is simply use the experiment that you were looking at. So the DNA is in a chromatin case in one particular cell. And what you do usually is that you look at the peaks, and then you only do the statistics inside the peaks because these are the only accessible regions. And one important issue is that there are actually two types of protocols which people use for doing the DNA. So we also wanted to evaluate what is the difference between these cleavers. So what we did here is we simply measured the Pearson correlation between the bias estimates of 60 libraries, and we can see that we can cluster them quite well by the type of how we measure. And this means that actually the naked DNase, which should be only capturing the cleavage bias, is actually different from the protocol-specific ones, and that the two protocols actually impose different bias. So this indicates actually there's more bias than only the cleavage itself. We can also evaluate this in a more practical approach, is that how does this work? So what we did is we used this bias estimate to simply correct the DNase signal by kind of renormalizing it. And what we show, at least here in combination of the method that we have previously proposed in the lab, hint, we see that if we use naked DNase, we improve significantly, but this is much more when we use the estimates based on the experiment itself, showing that one should really do bias correction. In a more practical perspective, so how does this affect the results? So these are, again, average profiles. So here, the cleavage bias. So this is one example of a factor that had an anti-footprint shape. And actually, after you correct, so which is this green line here, you see a more clear footprint. There's also other cases. For example, this was a motif that was in one of the main figures of the ENCODE paper I showed you before. And this was one case of a motif that wasn't shown before, and they were believed there was a de novo motif. But actually, the bias estimates also have a footprint-like shape. So after we correct, there is still a footprint, but this doesn't really reflect the sequence conservation. So this is likely to be a false prediction. So if you don't correct, you can really find things that don't make sense. So of course, what we did was we evaluated. Now we can evaluate all different methods. And here, I'm not going to go in details of everything, but I can skim out for some important results. So here is the AUPR values for 88 transcription factors. And if you see, this is what you can do only using the sequence. So you can do very poor. If you only kind of count how many reads you have in the peaks, which is, let's say, the simplest thing you could do, using DNase, you do much better. But then there are many other methods which try to do footprints, and they improve this task much more. And what was interesting to see that somehow site-centric methods had a worse accuracy than segmentation-based methods. And on a more complex analysis, which I not have time to show here, we saw that these three methods here were usually having better accuracy than all other methods. And it's also surprising because, for example, both Hint and DNase-seq try to do this cleavage bias correction. PIC does a very fancy smoothing, which also possibly accounts to remove this kind of artifact. Yeah, but one important information that we still saw was that there was still a large tail here in this distribution, meaning that for some reason it was hard to predict some of this to this prediction for some of the transcription factors. And actually, when we look at them, so usually the hard transcription factors were the same in, at least here in the top scoring methods. Yeah, so actually I just put this slide before because I saw in this, actually in this section before, there were a lot of people doing evaluation transcription factor binding using AUCs. And what I wanted to show here that you should not use AUC because you would look at negative and positives in the same way, but usually have much more negatives if you're looking, doing motive search. And that you see that in our cases, for all methods, there's much more, the uses are much more higher, so you're really overestimating your accuracy. And actually, all the deep learning methods, they do something around here, right? So open chromatin can really improve. But of course, it's not a fair comparison because they're only based on sequence. But coming back to the problem that we have is that the reason why we thought that some of the factors, the method wasn't working, was coming back to this transiency issue, right? So that for some factors, for example, CTCF is supposedly to have a large protection. So what we did was we simply proposed a statistic, which is just measuring the difference between the average values in your footprint or your bases in your motives versus the flanking regions. And if we do now here for very large, for much larger number of transcription factors, we see that there is a high association between the performance of the method, here shown by the AUC, versus the protection score. So the higher protection, of course, the less transient should be your factor. And actually, all the factors that people has reported before in the literature to be a short residence have a negative value, indicating that, so possibly one needs to use the protection score to kind of say, to kind of do a statement, which is, it's likely that for these particular predictions the DNase is not working. Okay, so with this I would like to conclude the talk. I have shown how actually DNase can really improve and push the current performance of transcription factors or cell-specific and active transcription factors. But for this, actually, one needs to really address experimental artifacts which you have in the data. And one particular outlook that we have is that there are now different alternative protocols, for example, attack-seq, also single-cell experiments, either in attack-seq or also in DNase-seq. And these experiments are also quite nice from the biological perspective because they are very simple to perform and also need fewer cells. So, for example, this is something that's still open computationally. And I also like to thank, in particular, Eduardo, who was the PhD student doing most of the work that I have shown here. Yeah, I'd like to thank you for your attention.